This conference will now be recorded.
There we go. Some people are just arriving now, a little bit later. So hello everyone, and welcome to today's webinar. This is the first webinar of the Ontario Invasive Plant Council's 2019 Winter Webinar Series. My name is Vicki Simkovic, and I'm the coordinator with the OIPC, and I'll be hosting today's webinar from Peterborough. Whether this is your first OIPC webinar or you're a repeat customer, welcome. This year marks the 6th year of the Winter Webinar Series, and we have a variety of really neat topics lined up for this year, from the management of dog strangling vine to restoring coastal wetlands in the Long Point region to hazardous plants and invasive species control and restoration on Kilally Island and the Canadian Health Inspection Agency's invasive plant program, as well as the Great Lakes Phragmites Collaborative and an overview of the Toronto Ravine Revitalization Study. The purpose of these webinars is to provide our network with the latest information on invasive plant control and management. We would also like to acknowledge and thank the Ministry of Natural Resources and Forestry for their financial support, which makes it possible to host these webinars. All webinars will be recorded, and you can access most of our previous webinar recordings and slides on our website at www.ontarioinvasiveplants.ca/webinars. We encourage you to access these resources for some of the most important information on issues relating to invasive plant management in Ontario. So before we begin today's webinar, all of you should have been placed on mute. So just look in the attendee box that your orange, there's an orange mic with a cross through it. Today's webinar will be about 45 minutes, and at the end we'll have some time for questions for about 10 minutes or so. So just put your question in the chat box and we'll get to it at the end. So we can get started. Today we're really excited to have Kathleen Ryan present an update on biocontrol for dog strangling vine. Kathleen Ryan is a forest entomologist working with Silvicon Limited since 2011. She's an expert in invasive forest species detection, ecology, and management. Current projects include the development of Hypena opulenta as a tool for management of dog strangling vine. Welcome, Kathleen. Thank you so much for being here, and you can get started in just a second. Okay.
Are you able to see my screen?
Yes, I can see your screen. Can you see?
Okay. Yes.
And you can hear me as well, it sounds like.
Yes.
Okay. So thank you all for joining me on your lunch hour today to listen to me talk about dog strangling vine biological control. I work with Hypena opulenta. Just a little teaser here. This is a picture of some of Hypena's handiwork in one of our field release sites from a couple of years ago. Before I get going, I'd like to thank the Ontario Invasive Plant Council and to Vicki specifically for organizing this webinar series and for inviting me to share our work here today. Before I get going, I just want to mention a webinar from about a year ago. Rob Boucher talked about biological control and talked about Hypena opulenta, and I'm going to kind of pick up where he left off, although I'll skew it more towards our company's role in this larger project. For those of you who haven't heard that webinar before, it's a really great overview of biological control and what it took to get to this stage of releasing Hypena in the field. How am I doing for speed there? I've got no audience to gauge response. I assume somebody's going to tell me if I'm talking too fast or too quiet. Okay. One thing I wanted to mention before I go too far is this is part of a large research consortium or research collaboration. Silvicon as a company is only looking at a small part of this bigger picture. We work most often with Agriculture and Agri-Foods Canada and University of Toronto researchers. We also work with York Region, who have been very supportive in assisting us with our field research and providing research sites. Some of the folks from Carleton University are working on this project as well. And University of Rhode Island and CABI in Switzerland did a lot of the, and they're still doing a lot of the work. They did a lot of the initial work, or all of the initial work, to identify Hypena as a biological control agent and go through the host range testing. And I'm going to present some of their results here today. I also want to mention the University of Rhode Island was very helpful in terms of us getting going with our rearing program as well. And you'll hear more about that later on in the talk. So today, this talk, I've got four main sections. One is a review of biological control, and this it touches on what Rob talked about last year, but I think it's important to set the stage. I will speak about Hypena biology, and after that, I'll talk about our research program, some of the projects that we're working on, and some of our results so far, as well as spend a few minutes talking about what our next steps are. So dog strangling vine, I'm sure if you're here, you're all familiar with it. Also known as pale swallowwort or Vincetoxicum rossicum. It's only one of the Vincetoxicum species here in North America. We also have Vincetoxicum nigrum or black swallowwort, and that's more common in the northeastern US, whereas pale swallowwort is more common here in Canada. But for the purposes of today's talk, we can lump them all in together because Hypena feeds on them both. So dog strangling vine or pale swallowwort is native to Eastern Europe. It's found in Ukraine and southwestern Russia. And there it's a forest species, so it's only found under closed canopy, and usually in small isolated patches, kind of unlike the situation here. And in Ukraine, where Hypena was identified in the first place, it's considered a rare plant. So that's in contrast to the situation here. So it was first recorded in Ontario in 1889, and it certainly has a broader habitat range here. So it tolerates a range of light conditions. It invades both natural and disturbed habitats, pastures, roadsides, forest margins. Here in North America, it has no effective natural limits on population growth. So there's very few herbivores that can feed on it, and none that can feed on it effectively to manage the plant, which is how we got to the biological control question in the first place. So this map is from the EdMaps program, and it's a map of DSV findings in Ontario. So DSV is found in Eastern Canada and in Ontario and Quebec specifically. It's become a significant problem in Ontario and Quebec over the past few decades. I mean, it's been here since 1889, but it was slow to get going. But in the past few decades, we've seen a very sudden increase in geographic range. And then within that geographic range, there's an increase in plant cover and plant density. One of the figures I found was that by early 2000s, which was about 100 years after it arrived, the range of dog strangling vine in North America was close to 40 million square kilometers. So it's pretty widespread problem. So the impacts of dog strangling vine, first of all, I should mention, and I know Stuart Livingston's on this webinar, this is a picture of one of our very first field release sites. And it was an agricultural field that was left abandoned for a few years. And the dog strangling vine took hold and took over everything. And it spread through the open area, and then it started to spread through the forest as well. And this is a pretty, it's on the extreme end of things, but it's not atypical. Which brings me to the impacts of dog strangling vine. So it forms a very dense monotypic cover, and it can be over very wide areas, and that can profoundly alter that invaded habitat. So it suppresses other plants. And our interest as a company is that it also suppresses tree seedlings and saplings, so there's a reduction in forest regeneration, and that's certainly something we see as a company. We're in sites that are very invaded by dog strangling vine. We don't really see that regeneration. So alongside that, there's a reduction in floral and faunal diversity. So with a reduction in plant species diversity, you tend to see fewer insects, and there's cascading effects in the food chain. From practical point of view, access issues makes it difficult for recreation and for forest operations as well. So management options. These are management options for invasive plants in general, and I'm sure you're all quite familiar with them. So first of all, do nothing, which has a risk in itself. Chemical and mechanical control are really the only options for dog strangling vine at this point in time, but they're rarely effective in controlling dog strangling vine unless you're really vigilant and do it multiple times per year and in successive years. Excuse me. Cultural controls, I'm not sure that we have many cultural controls that we're endeavoring in these situations. And basically, those are practices that make the weeds less competitive. Biological control is what we'll talk about today. And then the other point I want to bring up is integrated weed management, and that's something I'll touch on later. So how do you fit some of these tools together? So what is weed biological control? So biological control in general is using one organism to suppress another organism. So in this case, we're using an insect to suppress a weed. So classical biological control is the type of biological control where you're reuniting a natural enemy, in this case, Hypena, with a host, in this case, dog strangling vine, with the idea that you're trying to restore an ecological balance. So it's not a control strategy per se. We're not trying to eradicate dog strangling vine here. We're trying to restore some sort of ecological balance, similar to what would be found in its natural or its native range. It's cost effective at the spatial and temporal scale of the problem. So certainly in the early stages of a biological control program, it may seem more expensive, but if you consider the scope of the problem, it's fairly cost effective. The goal is that the organism is self-sustaining once it becomes established, and it has relatively low environmental impact, and that I'll touch on in the next few slides. So there's different stages of weed biological control programs. So this, you'll probably recognize this slide. This is Rob's slide from last year. So first of all, you need to know a lot about the species you're trying to control, and there's lots of literature out there, and I'm not going to talk about that. Then you go to the native range of that plant and look at the herbivores that feed on it there, or the natural enemies. Then there's host range studies to make sure that it's ecologically suitable to release here, and I'll touch on that. And then the petition for release, there's propagation and field releases. So we're sort of at stage 5 now, kind of moving into stage 6, which is looking at the establishment of the organism and the impact it has. And the final stage is redistribution and long-term assessment. And if any of you listened to Donna McKenzie's talk last year, that's the stage that she's at with the purple outstripe biological control. So dog strangling vine distribution in Europe, it's found, as I said, mostly in Eastern Europe. The Zootoxicum nigrum is found mostly in the Mediterranean, and there's another species, Hirundinaria, that's more pan-European. So when the overseas explorations took place, the researchers looked at, they did surveys in Western, Central, and Eastern Europe. This was back in 2006. To locate patches of this plant and to look at the herbivores that fed on Zootoxicum. These 5 agents were identified for further consideration. So there was a root feeder, a seed feeder, and 3 leaf feeders. And I guess the point I really want to make here is that in a natural ecosystem, there's many herbivores that feed on one plant and feed on different parts of the plant. Here we're just focused on one species, which is, you know, asking a lot of one species, but we're focused on Hypena opulenta. So it was chosen to focus on first because the original host range testing looked good, and also because it's multi-volting, so it goes through more than one generation per year. Which means there's more feeding impact over time. So the host range testing, First of all, Hypena was identified. It was collected in Donetsk in Ukraine. It went through a very rigorous host range testing to ensure that if it was released here, it was as safe as possible. So a long list of test plant species were identified by a technical advisory group. And it was tested against plants like, so related plants, environmentally and economically significant plants, threatened and endangered plants in the same family as Vincetoxicum. So those are things like the milkweeds, and plants that other Hypena species feed on. So there were 76 species that were selected for testing. And this was a no-choice testing, which is an extremely conservative approach. So the larva was caged with a plant, and they had a choice to feed on it or not feed at all. And what that does is it tests the fundamental host range of that insect. So it defines the absolute limits of its feeding range. And fortunately, it passed this host range testing with flying colors. So Hypena larvae only fed or only successfully developed on Vincetoxicum species. And then it went through the approval process, and I borrowed this slide from Rob as well. So first identified in 2006, went through fairly extensive host range testing between 2006 and 2011. The original release petition was submitted in 2011. There was additional testing that was completed and resubmitted in 2013. In September 2013, CFIA approved the release of Hypena opulenta in Canada. And that fall, there was the first releases of Hypena in the Ottawa area. APHIS, the counterpart of CFIA in the US, approved Hypena for release in 2017. So they're a few years behind us. Okay, Hypena, as I mentioned, is a leaf feeder. So it feeds on the leaf material, except for usually the mid rib of the leaf. It's a forest insect. It prefers closed canopy conditions. That's what we know from the literature, but that's also what we see when we release. It's multivolting, as I mentioned, and we do know that it goes through 2 generations per year in the parts of Ontario where we've released. So that was good news. It's native to plant hardiness zone 5 in Europe, which would be similar to the range of dog strangling vine here. So we're confident that it's a good climatic match. Okay, and here's the life cycle of Hypena. If you think back to elementary school biology, it goes through complete metamorphosis. So adult, egg, larva, pupa. So the adults, it's fairly inconspicuous. It's sort of a grayish brown. It's about 1 inch long, about 3 inches across when its wings are spread. And it has orangish shading on its hind wings. So the adults lay eggs on the underside of the leaf, and you can actually see them in the field if you flip over a leaf. They're kind of lightish yellow. According to the literature, a female that has fed on dog strangling vine through its lifetime produces about 300 eggs, although we don't see quite those numbers in the lab, but quite a few offspring. The eggs hatch. There's 5 larval instars, so 5 larval developmental stages. In this picture, you can probably see the larvae dangling from the underside of the leaf. So that would be a first larval instar, so 2-3 millimeters long. It's pretty hard to see. And of course, they get larger as they get older. They're about 2.5 centimeters at their longest. And this would be probably a 5th stage larva. They pupate in the litter layer or in the soil, or they kind of roll up dog strangling vine leaves and pupate there. And they overwinter as pupae. So as I mentioned, they go through 2 generations per year. So we see the first generation adults, it depends on the spring, but late May to mid to late June. And the second generation, we haven't quite seen the adults of that stage yet, but the caterpillars are around in August and into September. We certainly saw them into the middle of September last year. So it's fairly difficult to, so I'm going to flip back. It's difficult to actually see Hypena. It's pretty, they're fairly nondescript. They tend to be cryptic, I suppose is the best word. So they often feed on the underside of leaves. So unless you flip the leaf over, you don't typically see them. The adults fly at night, so unless you're stirring them up, you don't tend to see them either. What you may see though is larval feeding damage, and that's something, it would be great if everybody starts to think about that when they're out in the field and looking at dog strangling vine. It's not widely established now, but hopefully soon or before too long it will be. So this is something that's easier to see, the defoliation. So there's very few things that feed on dog strangling vine. Slugs sometimes, and sometimes you see a bit of mechanical damage. But if you see a lot of feeding damage, it's more likely to be Hypena. So there's kind of 3 categories of feeding you might see. The very young larvae, they feed on some of the cellular material, but they leave most of the leaf structure intact. But if you saw something patchy like that, you might want to flip over the leaf and see what's going on. As the larvae gets older, they feed more on the leaf, so you might see holes like that. And as they become older, you see that they consume most of the leaf material. They do prefer new leaves, but they do feed on older dog strangling vine leaves as well. And certainly in the lab, they'll feed on anything, including seed pods sometimes. Now, I've never seen that in the field, but they're pretty voracious eaters once they get going. And as proof of that, I got a bit of video here. So this is an older instar larva, and they just eat and poop all day long. Okay, are we still doing okay for talking speed and talking volume?
Yeah.
Okay,
Yes.
great.
That was perfect.
Great, thanks. Okay. My slide's not advancing here. There we go. Okay, so now I'm going to phase into the work we as a company have been doing. So we first became involved in this project in the spring of 2014. We're pretty excited about the project, and we were interested in the operational aspects. So those are things like developing a mass rearing program for Hypena. So how do you rear, thousands of them that we would need for a biological control project? I should mention this is in the scale of mass rearing programs. This would be on the smaller end of things. So we're looking at rearing thousands of caterpillars, not hundreds of thousands. So developing some good rearing methodology. We wanted to verify that so again, these are our objectives from 2014, and I'll talk through our results about that. We wanted to verify that it would reproduce and survive the winter in released areas. We wanted to look at the spread from those release points and dispersal. How far would it go? We wanted to examine the feeding impact on dog strangling vine. One of the things that we spent quite a bit of time on and still are is looking at effective implementation parameters. So how exactly would you release Hypena in the field? So what time of year would you release it? Like when in the summer? At what life stage? How many do you need to release to establish a population? And the other thing we were interested in is how exactly would we incorporate it into an integrated weed management program. So biological control is never going to be it's a tool in the toolbox. It's not going to be the silver bullet here. So how do you incorporate it with existing tools? So the first thing we needed to work on was to develop a rearing program. So we had some good ideas about what to do that, the folks at University of Rhode Island shared with us. And we used that as a starting point. And from there, we looked at ways to, maximize the efficiency of rearing, because it's a very labor-intensive endeavor. And how could we make it predictable? Like how could we, if we needed 500 larvae on a particular date, how would we do that? So we started off, or in the last few years, we've been working at a lab in Aurora. And last year we actually maxed out the space. So we reared 9,000 larvae and we had floor to ceiling Hypena. So no more room to grow there. So we're currently developing a larger space for next year. So we have 5 solid years of rearing data that we're using to guide our rearing program, and that's an ongoing project. And as much as you learn how to rear Hypena, as I found out, it always flows you. It always comes up with something new. So we're learning the troubleshooting as well. We're getting pretty good at that. So here's some pictures from our rearing program. We are rearing on live dog strangling vine and cut plants. And some of you may remember that Rob Boucher talked about artificial diet. We haven't gone that route at this time. So we grow potted dog strangling vine plants. We cage adults in with them. They lay their eggs on the plants. And then the larvae hatch, you can see on the right. And as they get older, they feed more heavily, and we can't keep them in potted plants. So we feed them cut dog strangling vine. If we're rearing them in the lab, we do rear a portion of them through for a lab colony, but we release as well. And these would be the stages we release at. So you can see this bin here on the right. So that would be what we would take out into the field. So that's the kind of the rearing portion of things in a nutshell. So I'm going to talk a bit about our field work, what we've done so far, some of the things we've found, and some of the projects we started last year. So what we found in previous years is that we can release at both larval and pupal stages. So for larval releases, the sites that we're focusing on are those with small to moderate sized patches of dog strangling vine. And by moderate sized patch, maybe it's as big as a room in your house, something like that. So what we do, basically we take the contents of that box you saw on the past slide and just fire it into a patch of dog strangling vine. These are dog strangling vine patches under closed canopy. And then we let them work their way through the patch from there and find a suitable place to feed. Typically, we've been releasing up to about 500 larvae in one of these patches. We're still working on what's that minimum number of larvae you need to establish a population, because it would be great if we could release fewer at each site so that we could then go on more sites. So this is a release site 2 weeks after release. So the sampling frame here would be centered on where that box contents were actually released. And all of this stringy stuff here is basically dog strangling vine stems and leaf ribs. So they've consumed most of the dog strangling vine. This is right at the release point. So it's certainly not uncommon to see 95% defoliation at that release patch. Not always, but often. And as you work away from that release patch, so you see the feeding density is a bit less. And you can see certainly feeding, you can see that they've moved, 3-4 meters away sometimes from that original release point. So this is a release site about 3 months after release. So we had released right in the center in early July, and this is September. So it knocked back the dog strangling vine and it didn't manage to regrow. This is from last year, so this is a site that we're monitoring to see what it does to the plant over time. What we often see is that the dog strangling vine turns yellow if there's been kind of moderate to intense feeding. The plant turns yellow and the leaves, it goes into early senescence. The leaves just fall off and it doesn't regrow. So we're not sure whether this is some sort of systemic effect or maybe just a generalized stress response of the plant. But either way, it's pretty good news. We also do pupal releases. So we put the pupae out in this box. You can see here, we hang them up from the tree. And then the pupae, the idea is that the pupae emerge As adults, they find each other, mate, and then go and find the dog strangling vine and lay eggs on them. Our thought was this would be great to use in sites with sparse dog strangling vine, so perhaps sites that are just starting to become infested with dog strangling vine, or where there's been chemical control and there's just some residual patches that have come back. So this was actually a project we had an intern do a couple of summers back. So she was tasked with developing a device that all the moths could get out of when they emerged. So she came up with some prototypes in the lab, and they worked great in the lab. And we tested them then at David Puddock's house. He's our boss. Just to field test them to make sure that they would be weatherproof and rodent proof and all of that sort of thing. And they were. And all the moths escaped, which is great. And as a bonus to this, he actually happened to know where the nearest dog strangling vine patches were to his house. And there's two of them. One of them was in an open area along the roadside, and we checked that out, no feeding damage. We checked about the time we would see feeding damage from the offspring of these pupae or the adults. So no feeding damage at all on that open patch, but then we went and looked at, there's a patch, again, about room-sized patch, moderate-sized patch, under forest cover, and it had feeding damage throughout that patch. So that was great. Not only can they get out of those boxes, they can find dog strangling vine, and that patch was 270 meters as the crow flies away from the release site. So they're pretty effective at finding those dog strangling vine patches, as you can expect they would be for something that only feeds on one species or one type of plant. In subsequent field releases, we've released this way in sites that just have those sparse dog strangling vine plants, like the one plant here and there, and they do find that as well. So that was a nice addition to our rearing or our release strategy. Another thing we do is monitor. So it's great to release, but we need to know that it's established, it's made it through the winter and it's established. So we have two methods that we use. One is light trapping and one is visual surveys. So this is actually a light trap that one of my colleagues rigged up from Canadian Tire and Dollarama parts, but it works great. It's not very specific to Hypena. It catches all kinds of species of moths. But as traps go, it's all we have at this point. So, and it does work. It does catch Hypena, so that's great. This is what it looks like at night. So we put those out kind of, again, depending on the spring, but late May to mid-June to monitor for that first generation of adults. And the other thing we do is visual surveys. So we have a look at the dog strangling vine, either kind of informally as we're walking through, or we do some formal surveys as well. And I'll talk about that in the next slide. All of the pictures here are from naturalized Hypena in field site. So we might see something like light feeding, which would be feeding like this sort of amount of the leaf gone and sparsely distributed through a patch. So that's light feeding. But we also see more moderate or heavier feeding where we see more of the leaf material consumed and throughout the plant and throughout the patch. So this is one of the things we looked at last year when I mentioned a more rigorous or thorough visual survey. So this was a site that there'd been a few releases at. So we were interested, and we knew Hypena had established there. So we were interested to know kind of where it was and how far afield it was. So in this site, we actually released larvae in 2015. We released 500 larvae there. We went back in 2016 and we knew it had overwintered because there was defoliation at this site again. As I mentioned before, there's very few things that defoliate Hypena, so we were quite confident it was Hypena itself. In 2016 as well, we did another research project over here. So we released actually close to 2,000 larvae that time. Excuse me. And we had light traps in that site in 2017 and 2018, and we knew Hypena was there. We were catching it on the light trap. But we had a summer student last year who we tasked her with walking transects through the site. So starting from the release patches and walking transects tight enough so she could see every single patch of dog strangling vine in there. And she mapped what she found. So she rated sites with Hypena feeding, or she also marked sites with no feeding. And here's what she found. So again, these are the two release sites. She found heavy feeding damage, and that would be damage like on the picture I showed you before. Sorry, I'm trying to go back to it, but it's not working. Anyway, so there's fairly heavy defoliation through the leaves and throughout the plant. So there's 4 patches of heavy feeding. There was several more patches with moderate feeding, several with light feeding. There was a few patches of dog strangling vine with no feeding damage at all. And then the green dots here were patches that there was something going on, but she wasn't sure whether it was feeding damage or from Hypena or something else. So anyway, the upshot of it is it's not only established here, but it's happily feeding and has certainly spread from those original release points. So it's gone a few hundred meters, regardless of which point it's spread from. So this is a site we're going to continue to monitor because we're interested in, do we start to see more of those heavy feeding areas, and have they found more patches? We expect that there's maybe a lag time with population establishment, so we're hoping to see this map fill in. I should mention that everywhere we found Hypena, it was all closed canopy. We didn't find it in open canopy areas at all, which fits with everything we knew about Hypena biology. Okay, my slides don't seem to be advancing here. Okay, there we go. So I had mentioned earlier, we're interested to know how Hypena fits in with an integrated weed management program, because in those sites with the vast dog strangling vine, I'm not sure that it's realistic to release Hypena on its own and hope for it to exert some amount of management, because that dog strangling vine has too far a head start, and I simply can't rear enough Hypena to make an impact in a reasonable frame of time. So one of the things I was interested in is, what if you knock it back with herbicide? But wouldn't it be interesting if you could establish a population of Hypena in the site before you herbicided it, and then it could clean up all those residual bits of dog strangling vine? So we had an opportunity at this site. The dog strangling vine was mapped. That's the yellow cross hatching here. And it was going to be treated in 2018. And so what we did was, went in before that, we established an area where we were going to release our Hypena, and we created a buffer. So all the areas outside that buffer would be herbicided, this patch would be herbicided, and we were going to release our Hypena in here. So we did some releases in June. So we released a few hundred Hypena at each spot there. It was sprayed in July, and they did a good job of conserving the buffer, because the dog strangling vine was still in good shape in the middle. We did some augmentative releases in July just because we happened to have some larvae left over. So this is a site we're going to continue to monitor to see if that's an effective method, but we're pretty excited about that. So we've been working on various research aspects of this project for a few years now, but now it's time for us to start thinking more operationally. So what we did last year was a couple of test operational releases. So one of our test cases was Kilally Meadows in the City of London. So this is an environmentally significant area. It's in northeast London. This is the North Thames River going through it. And this is an area, there's an ecological restoration plan underway for this whole park. And the objectives of the restoration plan, the emphasis is on invasive species management, especially of buckthorn and dog strangling vine. So our work fitted nicely with their overall management plan for the site. So there's more dog strangling vine than this, but this is a dog strangling vine that's mostly under forest cover. The blue sites had not been treated at all recently, and all these other, I think the red was going to be treated last year, the purple was treated the year before and the yellow before that. So there's a range of dog strangling vine density there. We did a walkthrough with the folks managing that site and the folks doing the ecological restoration plan. So we went in with the idea we were looking for closed canopy sites, small to moderate sized patches where we could release and to come up with a plan with the folks there. Excuse me. We did end up developing some additional release criteria after walking through the site. So one of them, there was a botanist with us that was really helpful conversation. We decided we would release at patches, or we would prioritize patches where there was good and native vegetation surrounding the patch, so that we could expect it would fill in any space left by dog strangling vine that was lost. We also considered this patch over here, but it's pretty close to the river, and this area floods quite frequently. So we were concerned about releasing an organism there that pupated on the ground and that maybe would be washed away in the spring. So we ended up doing sort of a multifaceted approach where we picked 3 sites for larval releases. So we released 500 larvae at each of those patches. And we released pupae at some areas where there was sparser dog strangling vine with those pupal release devices. And again, this is a project from last year, so this is a site we're going to go back to and monitor. I expect we'll end up, or I know we'll have to do augmentative releases because we want to try and build up the Hypena population a bit more quickly. So this is a site we'll work on this year. So I'm reaching the end of my talk here, but I want to just kind of remind everybody, this is a tool. It's a management tool. It's not an eradication strategy. If we release it, it's not going to get rid of all the dog strangling vine on the site. It's going to take multiple years. It may take augmentative releases. And really, the objective here, what we're looking to happen is it's going to reduce plant vigor. So there'll be less biomass. It'll reduce spread. It'll reduce seed set. So, I mean, that's what we all need to have in mind with this. Dog strangling vine has had a huge head start. It's going to be a while before we have that ecological balance we're looking for. So next steps for us, we're starting a small scale operational program beginning this year. So we're just looking to have 3 or 4 clients to start with. We feel we need to grow slowly to make sure that we manage the sites we're working on properly, and also to allow us time to ramp up our rearing program even more. We'll always have research aspects to this program. So a couple of the things I'm going to be working on this year, we're still working on that minimum population size, that what's the minimum number we can release to establish a population. And the other thing I'm really interested in is how do site factors fit in? So things like soil moisture, that sort of thing, how does that impact establishment? Okay, and that's it for me talking. So now I can answer questions.
Okay, we have 2 questions here. One about moderate feeding on a plant. Well, with moderate feeding on a plant, will the plant not be able to develop seed pods?
Yeah, they probably develop seed pods still. What we're hoping over time is that there'll be fewer seeds in those pods, or maybe we're trying to tap out its resources. Yeah.
Okay. How is the CFIA determining location of release sites like York Region?
The CFIA doesn't determine release sites. The CFIA said it's okay to release in Canada. We can release wherever we choose. They've approved release anywhere in Canada, uncaged release.
Okay. If field staff are noting Hypena feeding on DSV, is there an email or specific person we should be sending the data to?
Well, you can send it to me. My address is up here, and I can share it with. So Rob Boucher is kind of the overseer of this larger collaborative. So this is something that we would be sharing with the group more globally, but you can certainly send it to me. It's easier to put up my email right now, and I will share it.
Okay. After establishment in closed canopy areas, does Hypena spread to open field areas?
I doubt it. I don't know for sure, and that's something we're monitoring. I don't think so. It seems to be the more closed canopy areas are the ones that prefers. We'll certainly monitor that, and maybe I can come back and do a talk in a couple of years and the answer will be different, but I don't think so. There is another potential biological control agent, abrostula, and I think Rob mentioned it last year. It's found in open areas as well as closed canopy forests. So hopefully going forward, there'll be 2 organisms out there, and maybe abrostula has more utility in open areas.
Oh, that'd be great. And is Silvicon the only organization in Ontario currently undertaking rearing and release of Hypena?
We're the only ones undertaking the operational releases. So there's certainly research releases. So some out of Carleton, some out of UTSC. But in terms of actual treatment, yes, we're the only ones.
Are there some new sites planned for the new year?
Well, we are working on lining up some clients for this year, and it really depends on who those clients are, where the release sites will be. Our research sites, we've already determined them for next year. So if anybody was hoping for research sites, I'm sorry to disappoint you there. For research sites, we look at pretty specific, we look for pretty specific things. So we often tend to go back to forests we know really well, because we want to make sure the research design is as robust as possible.
Right. Okay. Question about so Vincent Toxicum changes soil chemistry to deter plant competition. Is there any indication that the stress of this biocontrol agent will affect that aspect of DSV's detrimental behavior?
Well, I'm not conducting that sort of research. That would be a larger scale, more longer term project. It's a really good question, but we're not at that stage. I think it would have to be knocking the plant back for several successive years before you might notice a difference. But again, that's kind of out of my scope here.
And what instar are the larvae generally released at?
I usually release a range of them.
Yeah.
It's interesting because the younger ones are more prone to predation, but then they have longer to feed, so there's a trade-off there. I don't release them really young, so not those L1s, and I don't release them when they're older, those L5s, because they're getting ready to pupate. But in the middle, I try and do some of each.
How long does it take to go through the first stage?
Sorry, can you repeat that?
Like, how long does it take to for all the instars to be completed into the pupal stage?
Right. So I can't remember the numbers off the top of my head. In the lab, it's about a month to go through the full cycle. Each instar, like the L1s, it's a couple of days, and then the older stages are a little bit longer.
Okay. How much does a series of pupae release? How much does a series of pupa releases cost to municipalities?
It really depends on the situation. I'm not going to disclose any numbers here. This is a discussion we can have offline, because it really depends on the situation. So we have to look at the site. It depends on how many we need to release. We're packaging this as a consulting service. We're not selling Hypena specifically. So we go in and advise about what would be the best approach for your site. So it's all kind of packaged together. I mean, Hypena, they're expensive to rear too, so that'll factor into the cost. I mean, we're talking a few dollars per insect. It's not cheap.
How many release sites are currently under assessment, and are they generally in the southern part of Ontario?
Well, we've so as a company, I'm just talking about our company, because there's releases only in the Ottawa area that I know less about. We've done a number of releases in the York region. We've done some closer to Uxbridge. We've done some in Simcoe. We've done some in London. And so what was the other part of that question?
Are they generally in the southern part of Ontario?
Yeah, right. So I guess that's generally south, although Ottawa, I'm not sure. It depends on how you're defining south, but that's kind of the range in general. So south, if you're, yeah.
Okay. Is there much mortality due to overwintering stresses at release sites that have been monitored?
There's those numbers are hard to get at. There is overwinter mortality. It's really hard to assess the population size. So we don't know how many we're losing to overwinter mortality. I expect that there's some, and it may relate to cold, but it'll also relate to moisture as well, I think.
Okay.
So that's a vague answer to your question. I don't know the numbers there is, though. That's something that we've got actually, Sandy Smith's postdoc student is looking at that as well. So hopefully he'll have some information about that soon.
And for closed canopies, what is the crown closure? So for example, 50 or 80%?
Yeah, so we're probably looking at, well, probably 70% and up would be ideal is what I would say off the top of my head. I do have those measurements that just escape me right now. Kind of depends on yeah. Let's go with that, 70.
Sure. I think we're at the end. Unless anyone has any other final questions. Oh, does the insect burrow into leaf matter to overwinter?
Yeah, so it pupates in the litter layer over winter or in the top layer of the soil. It doesn't go very far, but certainly in the litter layer where it's got a little bit of insulation.
Okay.
So one of the things you could look at actually as an aside, so not the overwintering ones, but the ones that pupate during the year, sometimes you can see them rolled up on DSV leaves that are still on the plant. So, if you see rolled up leaves on a DSV plant, it's worth investigating and see what you find.
We need a common name for Hypena to spark interest. What should we name it?
Yeah, we do. That's it. Okay, well, I feel like we should have a poll right here. That's a good question.
If anyone has any suggestions?
Dog strangling vine destroyer. The de-strangler. Perfect.
Are there any studies about the impact on species that might prey on Hypena?
There's not. There was an attempt to do that. There is predation on Hypena. I know that there was a student that attempted to do that, and I understood that is a challenging thing to study because predators don't want to be force-fed.
Yeah, it'd be hard to find them.
Yeah,
Is there any evidence? Yeah.
You were talking and I'm talking at the same time. I'll let you speak.
Oh, I finished with mine. Is there any evidence of root damage?
I haven't looked at the roots so far. I've just been monitoring above ground parts of the plant. It's a good question. It does need to be done.
Mm-hmm.
I was going to go just back to that previous question about the predation. So Hypena tends to be on the underside of leaves, like a lot of noctuid species. So they're hard to see. So if you're thinking about things like birds, I'm not sure how they'd be harder to find. So the predation I've seen, I think it's things like probably small mammals when they're pupating, but some of the homopterans seem to, like the ones with the sucking mouth parts, tend to feed on the larvae.
Are there any parasitic wasps, maybe?
Pardon?
Any parasitic wasps?
I don't know that they have any. They would have parasitoids, but I've not witnessed that. So I think when we get to the point where we're starting to recapture them from the field and bring them in, we'll get a better idea of parasitism.
I think we're at the end. Those were a lot of really great questions. Thanks, everyone. And yeah, thanks so much, Kathleen. That was a lot of great information.
You're welcome. Thanks for everybody for attending today.
Thank you very much.
Thank you.
Thank you. That was great.
Good.
Great, thank you.